we are going to discuss regulation of glycolysis, and with that we'll be able to illustrate some basic regulatory principles which are recurring motif in biochemistry. Glycolysis, as you all know, traditionally we start it from glucose, and by a series of steps it is converted to pyruvate. There are three irreversible steps. First one, catalyzed by hexokinase. Second, irreversible, catalyzed by the enzyme phosphofructokinase, converting fructose 6-phosphate to fructose 1,6-bisphosphate. And then the terminal step, where phosphoenol pyruvate is converted to pyruvate. So these three are the only, it'll rub it off. These three are only irreversible steps numbered as 3, 2, 1. So rest of the steps are reversible. And these three steps, the irreversible steps, are subject to regulation, which we are going to discuss now. The most important of all regulatory enzymes is phosphofructokinase, which we are going to discuss in detail. Phosphofructokinase, it is inhibited by ATP, but it is stimulated by ADP and AMP. It is inhibited by citrate by low pH, and as far as hormonal regulation is concerned, glucagon is inhibitor and insulin has got stimulatory influence. So we are going to now discuss the rationale behind them. Or just a minute, this one, the first one, it is subject to feedback inhibition by phosphofructose glucose 6-phosphate is inhibitor of this enzyme. So let us come to phosphofructokinase. These all are inhibitory influences and these all are stimulatory influences. We are going to see the rationale behind these now. This much is enough for passing your examination, but it is good if you can understand the rationale behind them, it will be good. So the first one, second one, as we said, phosphofructokinase, we are going to talk in details now. ATP, inhibitor of the pathway, you can very easily understand. Glycolysis is a ATP generating pathway. It generates ATP. And if ATP is already present in the body, in the cell, then there is no need for glycolysis to proceed. And that explains why glycolysis is inhibited by ATP. ADP, AMP का influence understand करने के लिए visualize a state where the muscle is contracting very fast, severe muscular exercise. तो यहाँ पर ATP is fastly it is being converted to ADP and AMP. What about energy need of the contracting muscle? More energy is needed. So glycolysis, more ATP is needed. So glycolysis must be stimulated. So therefore, in the contracting muscle, where ADP and AMP intracellular level is rising, so they will stimulate phosphofructokinase and therefore glycolysis. Citrate, as you know, it is the first intermediate of the TCA cycle. Increase in citrate concentration indicates that the fuel molecules are present. The mitochondrial fuel molecules are already present aplenty in lot. So already so many fuels are available, so there is no need for glycolysis to proceed fast. And one more thing, citrate, the first intermediate of TCA cycle, inhibiting the most important regulatory enzyme of glycolysis. So as a result of which glycolysis and TCA, they keep pace with each other. And then low pH, it is inhibitor of glycolysis because वी रिटर्न टू दैट कॉन्ट्रैक्टिंग मसल, तो वहां पर खूब फास्ट कॉन्ट्रैक्शन हो रहा है, lot of pyruvic acid, lot of lactic acid are being generated, and acidity rises, so pH falls. So this end product inhibition is there, indicating that no more of it is required. So glycolysis dampens with the falling pH. Now glucagon and insulin, glucagon and insulin. To understand this, let us see, let us return to fast, let us go to the fasting state. When a person is fasting, the blood glucose level is already dipping. The glucose level is going down. So at such points, the cell is using alternate fuels like fatty acid, ketone bodies, etc. for the energy needs. So glucose is precious, उसको प्रिजर्व करना है. सो अगर ग्लाइकोलाइसिस में चला गया ग्लूकोज, इट्स लेवल विल फर्दर फॉल डाउन. सो देयरफॉर ग्लाइकोलाइसिस विल slow down, and opposite is effect of insulin. So we have seen various effects now. Now we come to an important intermediate. Just rub it off now. An important intermediate which is a powerful stimulator of glycolysis. That intermediate is called fructose 2,6 bisphosphate. It is generated from fructose 6 phosphate only with the help of an enzyme called fructose 6 phosphate से ही बनता है. This is actually fructose 1,6 bisphosphate. This is fructose 2,6 bisphosphate, which is generated from fructose 6 phosphate by another enzyme, that is PFK2. So PFK2 generates this one, which is a powerful stimulator of phosphofructokinase. So this is actually just for nomenclature, we can call this as PFK2, this we can call as PFK1. Now an important point is that PFK2 is stimulated by fructose 6 phosphate. So this is an example of feed forward stimulation. I hope it's not becoming very complex, so you can eliminate. So you know, this point is getting a bit tough. So now we come to insulin. Now insulin is a stimulator of this enzyme PFK2. This is an example of covalent modulation. I'm going to not to going to talk much about it. Insulin is stimulator of PFK2. So insulin होगी, PFK2 constant activity बढ़ जाएगी. There will be raised level of fructose 2,6 bisphosphate, which will stimulate PFK1, and therefore the rate of glycolysis will be enhanced. Pyruvate kinase, there is nothing more I have to tell actually. Pyruvate kinase, it is inhibited by insulin. It is also stimulated by ADP and AMP, allosteric mechanism, and insulin or glucagon का same effect होगा. You know, I talked about covalent modulation. So these enzymes, they are active in the dephosphorylated form. So dephosphorylated form में अगर pyruvate kinase activated है, insulin causes dephosphorylation, as you all know. So therefore activity will increase of both these enzymes. So we have discussed the most important points about it. Now a word about feedback inhibition by this enzyme. Feedback inhibition by this of the first enzyme, that is hexokinase by glucose 6 phosphate. So इसका क्या फायदा होता है? फायदा इसका ये है, uncontrolled production of glucose 6 phosphate does not occur. अगर होना शुरू हो जाए तो उसका क्या नुकसान होगा? अनकंट्रोल्ड प्रोडक्शन ऑफ ग्लूकोज 6 फॉस्फेट, उससे क्या होगा? द अवेलेबल फॉस्फेट विल बी टाइड अप with the glucose. So the phosphate concentration will fall. And as you know, intracellular phosphate is very important for that phosphorylation reaction where ADP is converted to ATP. So feedback inhibition is there. So only that much of glucose 6 phosphate is produced what is required. The moment glucose 6 phosphate is converted to the next intermediate, इसकी concentration fall होगी, then only hexokinase reaction will occur. So that's all about it. We have discussed about three enzymes, the three regulatory enzymes, and the basic motive is it's an energy yielding pathway, glycolysis. So whenever energy charge of the cell is low, indicated by low ATP or high ADP AMP, or whenever fuel molecules are available, for example citrate. So ये high energy states हैं when no more energy or ATP is required. So they will halt glycolysis by various mechanisms. I have dwelt at length upon the allosteric modulations, and hormones, as I indicated, they act by covalent modulation, phospho, dephospho, etc. You can avoid these details. Thank you so much.